Hello and welcome back to our beginning look of our biomolecules. And just as a refresher, the four classes of biomolecules that we're going to be dealing with for the remainder of this chapter are listed there for you: carbohydrates, nucleic acids, proteins, and lipids. And if you remember from the last video, we said that not only is it our focus to understand what is a protein, what is a nucleic acid, what is a carbohydrate, what is a lipid, but our focus is also to try to understand how do these functional groups play into the biomolecule's ability to be reactive, to bond, and to work to maintain homeostasis. And so our goals here are twofold. Right? Yes, it's important to know what a carbohydrate is, but it's also important to understand how do we create complex carbohydrates like disaccharides and polysaccharides, things like starch, all right, things like glucagon. How do we create them, and what role do they play? And what are the processes that are involving if we want to break them apart? All of that is chemical bonding. A lot of that is dealing with covalent bonds. And all of that bonding is dealing with interactions for these functional groups. And so we're going to start applying all of this information and kind of building a network and warehouse of understanding and perspective, so that when we really get into the physiology, things start clicking and making sense that much better. And so with that said, let's start our look with carbohydrates. All right. Carbohydrate literally can be broken down into, this is not rocket science. This is definitely not complex understanding and complex science that's going on here. We can break this down into two parts: carbo and hydrates. Carbon and water. That's what a carbohydrate is. Right? And when we say that, we say that because there is a ratio between the number of hydrogens and the number of oxygen that is present within a carbohydrate. There is always a 2-to-1 ratio of hydrogen to oxygen. Right? But not only is there a 2-to-1 ratio of hydrogen to oxygen, there is always one carbon for every 2 hydrogens and 1 oxygen. Now, this is in your what we call monosaccharide type of carbohydrate, your simple sugar. All right. Once you start engaging in chemical bondings, these ratios kind of get skewed a little bit. But as soon as you go back to its basic structure, there is a 1 to 2-to-1 ratio. 1 carbon for every 2 hydrogens for every 1 oxygen. And so that's what you're seeing. That's what you're seeing right here. Right? The basic formula for a carbohydrate is 1 carbon for every 2 hydrogens for every 1 oxygen. Right? And the N on here is simply how many total. So for glucose, 6 carbon for 12 hydrogens for 6 oxygen. And so if we're following this, N here for carbon would be CH2O to the 6th, because that 6 is going to come over here, and you're going to have C6. 6 times 2 is 12. 12 hydrogens, and that 6 also goes to the oxygens, 6 oxygens. That is the basic formula for glucose. All right, that is the basic formula for glucose. Now, all of our biomolecules have a foundational level of organization, what we define as being a monomer. The monomer is the simplest level of molecular organization and still able to classify as the biomolecule. All right? So for carbohydrates, a monomer of carbohydrates, the simplest level of molecular organization that you can have as a carbohydrate and still be considered a carbohydrate, is a monosaccharide. Right? Saccharide means sugar. Mono means one. This is a one sugar molecule, right? Or this is a one molecule sugar. Now, if you take a monosaccharide and you join it with a monosaccharide, you have a disaccharide, two molecule sugar. All right. If you take a disaccharide and add another monosaccharide and another monosaccharide and another monosaccharide, or if you take a disaccharide and add a disaccharide and add a disaccharide and add a disaccharide, then you get a polysaccharide. A polysaccharide is three or more monomers of monosaccharides joined together. Right? And so what we need to do is we need to begin to understand, what do these look like, and how can we go through and identify these types of sugars, these types of carbohydrates, and what's the role of the functional groups? Remember, our main functional group that we're really concerned with here are hydroxyl groups. Right? Remember what a hydroxyl group looks like. Right? That's the OH. And so that's what we're going to be going into next. So let's go through and look at the monosaccharides. What are some of the basic characteristics of a monosaccharide? Well, there's two varieties of monosaccharides. We can have 5-carbon sugars. We can have 5-carbon sugars. Hold on one second. Let's go to magenta. You can have 5-carbon sugars. And these 5-carbon sugars are actually seen, they're found within nucleic acids. And so these sugars actually define RNA and DNA. Right? RNA stands for ribonucleic acids. Why? Because it contains the sugar ribose. DNA is deoxyribonucleic acids. Why? Because it contains the sugar deoxyribose. And this is paramount. This is critical in our understanding of bonding and how stable DNA is versus RNA. We're going to get into this a lot more. I get super excited over this kind of stuff. But real quick, right, pay attention to this. Deoxyribose. This is a sugar that is without one oxygen. Deoxyribose. Deoxyribose is a sugar that is missing one oxygen. That is the only difference between ribose and deoxyribose. Right? Look down here. Here they are right here. And by the way, for the exam, you do need to be able to distinguish these two molecules. You need to be able to look at these molecules and say, yep, this is ribose, or yep, this is deoxyribose. All right? And here's the thing. So we count carbons. Right? We always start on the right and we move to the left. So and wherever you see a point, right, with no atom, that's a carbon. And so this is 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. All right, so this is a 5-carbon sugar. All right, pentose. All right. Same thing, that's deoxyribose. Same thing with ribose. 1, 2, 3, 4, 5. And when you look at deoxyribose and you look at ribose, they look identical, except this is what we call the 2' carbon. All right? Look at the 2' carbon. Look right here at the 2' carbon. Here, it's got a hydrogen atom that's attached. Here, it's got a hydroxyl group. It's got a hydroxide attached, an OH. OH here, no OH here. Other than that, these two molecules are identical. Go back to the name. Deoxy, without oxygen. Right there. That's where deoxyribose gets its name from. It gets its name because on the 2' carbon, it is lacking a hydroxyl group. There's only a hydrogen. It's missing an oxygen. And as we will discover when we get into nucleic acids, that missing oxygen is the difference between your DNA being stable and your DNA breaking down, just like ribose or just like RNA does. And so we're going to get into that a little bit more as we get into nucleic acids. And then again, we'll revisit that in chapter 3 when we talk about things like protein synthesis and DNA replication. All right, so that's one group of monosaccharide. The other group of monosaccharide is one that you're more familiar with, and that's your 6 carbon or your hexose types of sugars. Right? So hexose are sugars. The ending OSE always means a sugar. Notice over here for the pentose, because it's 5, it ends in OSE. All right. Ribose ends in OSE. All right. Deoxyribose ends in OSE. Right. OSE always means sugar. Right. Hex means 6. And so this is a 6 carbon sugar. Right. And there's 3 basic ones that we encounter almost on a daily basis. Glucose, fructose, and galactose. All right. Glucose is what we typically identify with blood sugar. All right. Fructose, again, is almost identical to glucose, with the exception of you got this additional group right here, right? And everything else tends to be flipped. Right? But these guys are almost mirror images of one another. And then you've got galactose as well. Now, these are all 6 carbon sugars. You need to know that glucose is an example of a monosaccharide. It's a 6 carbon sugar. You need to know that fructose or fructose is a monosaccharide. It's an example of a 6 carbon sugar. You need to know that galactose is a 6 carbon sugar. Right? It's a monosaccharide. You do not need to know their chemical formulas. In other words, you don't need to be able to look at them and identify them from a picture. Deoxyribose and ribose, you do. Right? Deoxyribose and ribose, you absolutely do. Right? And so these are your monomers of carbohydrates. Right? You've got 6 carbon sugars, glucose, fructose, and galactose, and you've got 5 carbon sugars, deoxyribose and ribose. We can take these individual monomers, these monosaccharides of carbohydrates, and join them together. We can take glucose and attach it to fructose. We can take glucose and attach it to galactose. We can take glucose and connect it and join it to glucose. And when we do, we now have 2 molecules that are forming our carbohydrate. Right? In other words, we have a disaccharide. It is a 2 sugar molecule. Right? Sucrose is an example of a disaccharide. It is glucose and fructose joined together. By the way, sucrose is table sugar. Right? So you sprinkle this on your cereal, you put it in your coffee, you add it to red spaghetti sauce, you add it to cakes and pies and pastries. We add sugar to almost everything. Right? When you're using that white granulated sugar, it's sucrose. Right? It's a disaccharide. Maltose is a sugar that is actually produced through starch digestion. Right? So starch digestion starts in the mouth. Right? You'll learn about this when you get into the digestive system in A&P2. But as starch is broken down into simpler and simpler molecules, you will go ahead and you will create maltose. Now, glycogen, right, which is a stored version of starch. Right? Starch is a plant polysaccharide. Right? The analogous version of that in the human is glycogen. So glycogen and starch are the same thing. Starches are found in plants. Glycogen is found in humans. Chitin, Chitin. Chitin is the exact same thing as starch, and glycogen, just an insect. So if you've ever found the outer skeleton, the shedded skeleton, the exoskeleton of an insect, that's made of chitin. Right? And so we can go through and we can keep going on as far as what these analogous structures are. But know that when you break down starch, you get maltose, and then maltose can be broken down into glucose. Right? And then we've got lactose, which is galactose and glucose that are joined together. And lactose, we know, is a milk sugar. Right? Now, interestingly enough, there's an enzyme that we produce. Hold on one second. There's an enzyme that we produce called lactase. Right? That's an enzyme. And lactase is an enzyme that breaks down lactose. It does that in the small intestines. It's in high levels of production when you are an infant. Why? Because you're nursing. You're drinking milk, and you need to be able to break down the lactose. All right? The problem is, as you age, you decrease the production of lactose. Right? You decrease the production of lactase, which means your ability to break down lactose also decreases. Now, this happens at varying stages of individuals' lives. Right? Up until about 4 years ago, I could eat as much ice cream and pizza and everything else as I want. I'm now starting to notice with things like ice cream, I get that indigestion feeling. I know my lactase levels are starting to decrease. Other people, by the time they're in their teens, the lactase is gone. It's history. Out of there. Sorry. All right. It's done. And so everybody loses or starts to have decreased production of lactase at different times of their lives. Why? Because we are the only animal. Yes, we are an animal. Right? We are the only animal on the face of the earth ever in existence to continue to consume milk past infancy. That's it. You're not designed to continue to drink dairy product and eat dairy product past weaning, right? Past the infancy stage. And so, evolutionarily speaking, we don't need it, so we lose it. So technically, if you are lactose intolerant, you're normal. The diseased person, the person that has a condition, is the person that can still digest lactose, because they're the abnormal ones. They're producing something that your body shouldn't still be producing. We have to flip that on its head. Lactose intolerance is normal. You're not abnormal. You don't have a disorder. Those of us that can still break it down, we're the abnormal ones. We are the diseased ones. Because it's not supposed to happen. Now, what ends up happening? Well, you're not producing lactase, right? And the lactose now passes from the small intestines into the large intestines. And in the large intestines, there is bacteria. Now, not all bacteria can ferment or break down lactose. In fact, enterobacteria can, and E. coli can. And out of those two, E. coli is highly aggressive in being able to ferment lactose. So much that it actually grows green. It's a metallic green when you grow it on a specialized auger plate, right, for bacteria, for E. coli. And so it grows this metallic green sheen. You load it with lactose, and does it ferment it? Right? And if it ferments it, it grows. It's a pretty shiny metallic green color on it. Well, in your stomach or in your large intestines, E. coli starts breaking down the lactose because you're not producing lactase, and this needs to go. So you're feeding your colon E. coli. But bacteria produce gas as a byproduct of fermentation. And that gas builds up in the large intestines, and it backs up into the small intestines, and you get bloated, and you get cramps, and you get abdominal pain because you've produced all of this gas through the use of fermentation with your E. coli in your large intestines because you're not producing lactase because you're normal. You're abnormal because you keep drinking the stuff and eating and consuming dairy. But you are completely normal because you're not producing lactase any longer. Right? So when someone says they are lactose intolerant, congratulate them for being normal. Right? And reflect on how you are actually the abnormal one, right, within that group. Now, oh God, what does this have to do with anything? Okay, well, side. I just took the struggle bus and completely detoured you guys off the main path. So I got to get the struggle bus back onto the main highway as the bus driver and get this going. So disaccharides actually are joined together through a process called dehydration synthesis. Process by joining two monomers together. Right? Now, we joined by joining two molecules, two monomers together through the removal of water, dehydration, right? H2O out. We're removing it. And when we remove water, we synthesize. Dehydration synthesis. We're creating a new molecule through the removal of water. Right? Through the removal of water. All right. Now, where does that happen? Well, look right here. Right? This is the result of dehydration synthesis. This is the result of dehydration synthesis. This is the result of dehydration synthesis. Right? And what we will see on the next slide is how this happens in a little bit more detail. And so that is kind of where we are heading with this. And so let's go ahead and let's start to look now at this process of how we exactly join these monosaccharides to be able to start creating disaccharides and polysaccharides. I will say that once we learn the basics of this, there's going to be a lot of repetition as we move through the other biomolecules. All right. So this here is just a summary for you as far as what is the difference between a monosaccharide, a disaccharide, and a polysaccharide, which again, poly just means long chains of either monosaccharides or disaccharides, or a combination of di and monosaccharides that have been linked together. So how do we do this? How do we progress ourselves through this process? How do we go from being a monosaccharide to a disaccharide to a polysaccharide? So let's look at that. Again, as I've already alluded to on the earlier slide, that the process that we use to move through becoming a disaccharide or a polysaccharide is this process of dehydration synthesis. All right. This idea that we will remove water to produce a more complex molecule. All right. So the name defines it. Think of it in those terms. Well, what does this mean from an atom perspective? What does this mean from a molecular perspective? Well, the way that we create this molecule water to form this bond is by, as it's described here, we take a hydrogen atom from one monosaccharide, and we take a hydroxyl group from another monosaccharide, and we create that water. Right, so let's look down here. All right, here's a monosaccharide. Here's a monosaccharide. All right, doesn't matter what it is. It could be glucose, it could be fructose, it could be galactose. It doesn't matter. Right, it's a monomer. It's a monosaccharide. All right, this is a monosaccharide. All right, so we're going to remove from monosaccharide number 1, we're going to remove a hydroxyl group. And from the second monosaccharide, we're going to remove a hydrogen atom. Right, well, what have we done? We've removed 2 hydrogens and an oxygen. H2O. Right, and when we do that, let's look over here. Here's that water molecule leaving. We create a new bond that with their leftover oxygen. So this oxygen right here is still remaining. And it's this oxygen that creates a bond that now links monomer 1 to monomer 2. This bond that is now created from removing a molecule of water from 2 hydroxyl groups is called a glycosidic bond. All right, this is what we call a glycosidic bond. A glycosidic bond is formed between 2 monosaccharides where a hydrogen atom and a hydroxyl group is removed from 2 hydroxyl functional groups. All right, now that's a lot. That's a mouthful. A glycosidic bond is formed between 2 monosaccharides where a hydrogen atom and a hydroxyl group is removed from basically 2 hydroxyl functional groups. All right, so this whole hydroxyl group is gone, and only this hydrogen is gone from this hydroxyl group. And again, what is left over is this oxygen molecule that is now linking the 2 monosaccharides to create this glycosidic bond. All right. This next image here will kind of give you a little bit different of a perspective there. And I apologize for all of the writing over. Let me see if I can do away with that writing here real quick. Oh, there we go. All right, so now we can see this a little bit easier. So check this out. All right. Here's glucose. Here's fructose. All right, we're going to make sucrose. So using that definition from before, here are our 2 hydroxyl groups, right there and right there. Here's the hydroxyl group from the monomer of glucose. Here's the hydroxyl group from the monomer of fructose. All right. From these 2 hydroxyl groups, we are going to remove the whole hydroxyl group and a hydrogen from this hydroxyl group to create the H2O. H2O. All right, and what's left over? Well, once again, that oxygen is left behind. And it's that oxygen right here that now forms the glycosidic bond. Right, that is a glycosidic bond. Right, we only find glycosidic bonds associated with carbohydrates. Right? And every time we add a monomer, every time we add a monosaccharide onto this chain, we are going to do dehydration synthesis, resulting in a glycosidic bond. All right. So polysaccharides, just kind of real quick, and we've kind of already have looked at this. All right. Polysaccharides are simply a monosaccharide attached to a monosaccharide attached to a monosaccharide attached to a monosaccharide attached to a monosaccharide, so on and so forth. All right? So we keep doing this dehydration synthesis, getting this very large complex molecule such as glycogen. Right? Again, glycogen, it's stored in the liver and it's stored in skeletal muscle primarily. There's some other areas within the body that it's stored in, but this is an energy reservoir. As we're going to learn in Chapter 3, glucose is what is needed in order to drive cellular respiration and to create this golden molecule of energy called ATP, adenosine triphosphate. Glucose is key in that. So we keep on reserve these large molecules of glycogen because they are glucose. It's what they're made of, right? And I made reference to this that in humans we call this glycogen. Right? In plants we refer to this as starch or cellulose. In insects we refer to it as chitin. Right? CHITIN. Right? In bacteria, we call it something else, peptidoglycan. It's all the same thing. It's all glucose that's been rearranged through this process of dehydration synthesis. So we get this very large and complex molecule, and now the question becomes, how do we break it down? What's the opposite of dehydration synthesis? If we're doing dehydration synthesis to create this complex molecule, there's got to be something that's opposing it or that is opposite to it. And the answer is you are correct. There is something that is opposite to this. By the way, this is just an overview of how did we get there? How did we get to this polysaccharide? And it comes down to dehydration synthesis. There's the glycosidic bond. There's the glycosidic bond. Here's the glycosidic bond. So we repeatedly do dehydration synthesis. The opposite of that? Well, if we removed water to build the bond, then the opposite of removing water to build the bond would be to add water. To break it apart. And so the process that we do is hydrolysis. Right? We add H2O back into the molecule to break the glycosidic bond. So here's our glycosidic bond. Right, here's monomer 1, here's monomer 2. Now we're going to add that water back in. And when we add the water back in, we've got H2O. Well, OH comes back over here and reestablishes this with the primary carbon. All right. And the leftover hydrogen comes back over here and attaches to the oxygen that was once forming the glycosidic bond to reestablish the hydroxyl group here. And so you come back to having these two hydroxyl groups that are established. And let me kind of go back. So if we look at this screen here, here's, once again, Here's the glycosidic bond. Right? Here's the oxygen. This is the 1' carbon. Right? This would actually be the 5' carbon over here. Because if we count carbons, let's get you used to doing that. Let's have you think about counting carbons. All right. Carbon 1, 2, 3, 4, 5, 6. Carbon 1, 2, 3. Carbon 4. Hold on one second. Carbon 4. Oops, I lied. Carbon 1. Carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. All right, I lost a carbon along the way. Sorry about that. And so this glycosidic bond is formed between the primary carbon and the 5th carbon over here. Well, when we add that water back in, right, remember, we're dealing with an OH plus an H. And so the OH comes here, the H comes over here, right, and you restore the hydroxyl groups, right, that you once went ahead and broke down from before. All right. So the opposite of dehydration synthesis is synthesis, where we reestablish these connections. And this here just kind of is a summary table for you to kind of look at, and again, to summarize what is happening during both dehydration and hydrolysis. So a lot for you to take in. Not only are you learning what a carbohydrate is and how it behaves, but you've got some new processes that you are also considering. Right? Dehydration, synthesis, and synthesis, and how we use that to identify the functional groups and to go ahead and create more complex molecules and to break complex molecules down into their monomers. So a lot to review, a lot to digest. I'm going to leave you to that. And if you have questions, make sure you always reach out and ask. And as you have become accustomed to, I'll catch you on the flip side.